Let's take a journey together tonight. A gentle journey into the world beneath our feet and all around us. A world that exists in the quiet spaces of the forest floor, in the damp coolness of fallen logs, in the shadows where sunlight filters through ancient trees. Picture yourself walking through a peaceful woodland on a calm evening, when the day's heat has given way to a gentle coolness, when the light takes on that golden quality that makes everything seem softer, more magical. The forest floor is carpeted with fallen leaves, layer upon layer of them, creating a soft cushion beneath your feet. And there, emerging from this blanket of decay and renewal, you see them, mushrooms in their infinite variety, from tiny caps no bigger than your fingernail to broad shelves spanning the width of old tree trunks. Tonight, we're going to explore these remarkable organisms together, but we're going to do it slowly, peacefully, letting each fact settle over you like a soft blanket, letting the wonder of the fungal world wash away the tensions of the day. There's something deeply calming about mushrooms, about their quiet existence, their patient growth, their essential role in the slow cycles of the forest. They don't rush. They don't demand attention. They simply are, doing their work in the shadows, connecting, decomposing, nurturing, existing in a state of perpetual stillness and constant change all at once. So settle in, let your breathing slow, and join me as we discover the most peaceful, fascinating, and sleep-inducing facts about these extraordinary organisms. The ancient origins of fungi. Long before humans walked the earth, long before the first flowers bloomed or the first birds sang, fungi were already here, already thriving, already doing the quiet work of transformation that would make so much of life possible. The story of fungi stretches back so far into the depths of time that it becomes almost dreamlike in its vastness. A story measured not in years or centuries but in hundreds of millions of years. A story that predates most of what we think of as the living world. Scientists have discovered fossil evidence suggesting that fungi have existed for at least 1.2 to 1.5 billion years. Think about that for a moment. Imagine a world so ancient that it bears almost no resemblance to the world we know today. Imagine oceans without fish, land without plants, a planet that would seem utterly alien to our eyes. And yet, even then, fungi were present, microscopic organisms living in the water, moving through their environment with tiny whip-like structures called flagella, the ancestors of all the mushrooms we see today. Research published recently has revealed fossils of fungi from rocks dated between 715 and 810 million years old, discovered in the Democratic Republic of the Congo. These ancient remains showed the characteristic branching filaments of fungal mycelium, those thread-like structures that are the main body of a fungus. When researchers examined these fossils with sophisticated microscopic techniques, they found traces of chitin, the tough compound that makes up fungal cell walls, confirming that these were indeed fungi, living in what was likely a transitional environment between water and land, in ancient lagoons or coastal lakes where the world was slowly changing. The preservation of these fossils is itself a kind of miracle, a moment frozen in time when these delicate organisms were trapped in sediment, compressed over eons, transformed into stone while somehow retaining enough of their structure that scientists millions of years later could identify them, could see in their patterns the same basic architecture that exists in fungi today. It's a connection across time so vast it's almost incomprehensible, and yet there it is, written in stone, a testament to the incredible persistence of fungal life. For much of their early history, fungi lived in water. They were part of the ancient aquatic ecosystems that covered much of the early Earth, moving through the water column with their flagella, feeding on whatever organic matter they could find. But then something remarkable happened. Around 400 million years ago, as plants began their own transition from water to land, fungi made that journey too. And this transition, this movement from an aquatic to a terrestrial lifestyle, required profound changes in how fungi obtained their nutrients and survived in this new, drier environment. The earliest known mushroom fossils, the actual fruiting bodies that we would recognize as mushrooms, date back to about 90 million years ago, from the Middle Cretaceous period. These ancient mushrooms, preserved in amber like something out of a fantasy story, belong to an extinct species called Archaeomarasmius legeti. Picture these mushrooms growing in the age of dinosaurs, in forests populated by creatures that would seem mythical to us now, yet producing the same basic structure that mushrooms produce today. A cap, a stem, gills underneath for producing spores. But mushrooms themselves are just the tip of the iceberg, the visible fruiting bodies that emerge briefly to release spores before withering away. The real organism, the main body of the fungus, lives hidden in the substrate, spreading its network of fine threads through soil, wood, and organic matter. This mycelial network can persist for years, decades, even centuries, growing slowly and steadily, breaking down complex organic materials, forming relationships with plants, existing in a state of quiet persistence that asks nothing of us, demands nothing of our attention, simply continues its work year after year. The transition from water to land wasn't just about surviving in a new environment. It was about transforming that environment. Early terrestrial fungi played a crucial role in breaking down the first dead plant material on land, helping to create the first soils. Before fungi, when plants died, they simply accumulated. There were few organisms capable of breaking down the tough structural materials that plants had evolved, the cellulose and lignin that made them sturdy enough to stand upright in air rather than floating in water. Fungi evolved the enzymes necessary to decompose these materials, and in doing so, they became the planet's first and most important decomposers on land, recycling nutrients and making them available for new plant growth. This created a feedback loop, a partnership between fungi and plants that would transform the entire planet. Plants could grow, die, and be broken down by fungi, releasing their nutrients back into the soil where new plants could use them. This cycle, this constant turning over of organic matter, created deeper soils, more fertile environments, conditions that allowed plants to grow larger and more diverse. And as plants evolved and diversified, so did fungi, developing new strategies, new forms, new ways of obtaining nutrients. Some fungi became specialists in forming intimate relationships with plant roots, developing into what we now call mycorrhizal fungi. These fungi wrap around or penetrate plant roots, forming structures that dramatically increase the plant's ability to absorb water and nutrients from the soil. In return, the plant provides the fungus with sugars produced through photosynthesis. This mutualistic relationship is so ancient, so fundamental, that it's estimated to have evolved at least 400 million years ago, roughly the same time as the first land plants were establishing themselves. The evidence suggests that this partnership between fungi and plants was not incidental but essential. Early land plants likely could not have survived without their fungal partners. The soils of early land were thin, nutrient-poor, lacking the rich organic content of modern soils. without mycorrhizal fungi to help them extract every possible bit of nutrition from this challenging environment, these pioneer plants would have struggled to survive. Fungi, then, were not just present during the greening of the land, they were instrumental in making it possible. As the millions of years passed, fungi continued to evolve and diversify. Different lineages developed different strategies for survival. Some remained decomposers, breaking down dead organic matter. Others became parasites, feeding on living plants, animals, or even other fungi. Still others maintained their mutualistic relationships with plants, co-evolving with them as both groups adapted to new environments and conditions. Around 250 million years ago, after a massive extinction event that killed most life on Earth, fungi experienced what scientists call a fungal spike. In sediments from this time, fungal spores make up nearly 100% of the fossil record in some locations, suggesting that fungi became the dominant form of life in the aftermath of this catastrophe. While the exact interpretation of this evidence is still debated, it paints a picture of fungi thriving in conditions where other life struggled, breaking down the enormous amounts of dead organic matter left behind by the extinction, slowly but steadily preparing the world for the recovery of life. Later, around 66 million years ago, after the extinction event that ended the age of dinosaurs, another fungal bloom occurred. Again, in the wake of catastrophe, fungi flourished, doing what they do best: decomposing, recycling, transforming death back into the building blocks of life. It's a sobering thought, perhaps, but also an oddly comforting one. No matter what happens, no matter how dramatically the world changes, fungi persist, patient and persistent, waiting in the soil, ready to begin again their slow work of transformation. The lineages of mushroom-forming fungi that we see today began to diverge and diversify during the late Carboniferous period, around 300 million years ago. By this time, forests dominated much of the land, creating vast amounts of woody material that needed to be broken down. Fungi evolved increasingly sophisticated enzyme systems to tackle this challenge, developing the ability to break down lignin, one of the toughest and most resistant materials in nature. Some of these evolutionary innovations happened remarkably recently in geological terms. For instance, the ability to form the typical mushroom shape with its cap and stem and gills evolved multiple times independently in different fungal lineages. This suggests that this particular structure is especially good at what mushrooms do: producing and dispersing spores efficiently. The elevated cap protects the spore-producing surface underneath, while the arrangement of gills or pores maximizes the surface area for spore production, allowing a single mushroom to produce millions or even billions of spores in just a few days. As we trace this ancient history, from those first aquatic fungi swimming with their flagella over 1 billion years ago to the enormous diversity of fungi we see today, we're tracing a story of patience, adaptation, and persistence. Fungi don't rush. They work on timescales that make human lifespans seem like fleeting moments. A fungal colony can grow for decades, spreading its mycelium slowly through the soil, forming connections, breaking down materials, existing in a state of quiet readiness. And when conditions are right, when the temperature and moisture are perfect, it can produce a mushroom in a matter of days, a brief flowering of reproductive effort before returning to its patient underground existence. There's something deeply calming about this perspective, about seeing ourselves as part of a story so much larger and longer than our individual lives. The fungi growing in the forest near your home are connected, through an unbroken line of descent, to those ancient aquatic fungi of 1 billion years ago. They're still doing essentially the same work, still breaking down organic matter, still forming relationships with plants, still producing spores and sending them out into the world. They're a living link to the deep past, a reminder that life goes on, that the cycles continue, that there's a rhythm to existence that's much older and slower than our daily concerns. The hidden underground networks, beneath every forest, under every meadow, spreading through the soil in patterns too vast and intricate for the eye to see. There exists a network of fungal threads that scientists have come to call the Wood Wide Web. This name, a playful reference to the World Wide Web that connects our computers, hints at something profound: that forests are not simply collections of individual trees competing for resources, but rather communities connected by vast underground networks of fungal mycelium, networks through which nutrients, water, and even chemical signals can flow. The mycelium of a single fungus is made up of incredibly thin threads called hyphae, each one typically only a few micrometers in diameter, far thinner than a human hair. And yet, despite their delicate nature, these hyphae are remarkably tough and persistent. They grow through soil particles around rocks, through decaying organic matter, branching and rebranching, forming a network of almost unimaginable complexity. Scientists have estimated that in a single teaspoon of healthy forest soil, there can be several miles of fungal hyphae, all of it belonging to multiple different species, all of it actively growing, exploring, seeking out resources. When we scale this up to the level of an entire forest, the numbers become almost impossible to comprehend. Under a single square meter of forest floor, there can be thousands of kilometers of fungal hyphae. This network permeates the soil at every level, from the surface layers rich in fresh organic matter down into the deeper mineral soil. It wraps around roots, penetrates dead wood, spreads through the leaf litter. It is everywhere and nowhere, invisible to our eyes but fundamental to the functioning of the entire ecosystem. These networks don't grow randomly. Fungi are actively exploring their environment, seeking out resources, responding to chemical signals, growing towards some things and away from others. When a hyphae detects a source of nutrients, perhaps a cache of organic nitrogen or a root exuding sugars, it grows more densely in that direction, concentrating its efforts where the rewards are greatest. This creates zones of higher and lower fungal density, patterns within patterns, a landscape of opportunity and effort playing out in the soil beneath our feet. One of the most remarkable aspects of these fungal networks is their role in connecting plants. Many species of fungi form mycorrhizal relationships with plant roots, and a single fungal individual can connect to multiple plants simultaneously. A fungus colonizing the roots of one tree can send its hyphae out through the soil to colonize the roots of neighboring trees as well, creating a physical connection between plants that can span considerable distances. Through this connection, the fungus acts as an intermediary, a living bridge between plants that might otherwise have no direct interaction. Research has shown that substances can move through these fungal networks from one plant to another. In laboratory and field studies, scientists have labeled certain plants with isotopes, special forms of carbon or nitrogen that can be tracked, and then watched as these labeled elements appeared in neighboring plants connected by the same fungal network. This demonstrates that transfer is possible, that the mycelial network can act as a conduit for resources moving between plants. The mechanisms of this transfer are still being studied and debated. Some resources might move directly through the fungal hyphae, traveling through the cytoplasm of the fungal cells from one location to another. Other resources might be released by the fungus into the soil around one plant's roots and then picked up by another plant's roots nearby. The fungus might not be deliberately facilitating plant-to-plant transfer, but rather acting as an intermediary that happens to connect multiple plants while pursuing its own needs. What's clear is that these networks are complex and that multiple processes are happening simultaneously. The fungus is trading with plants, receiving sugars in exchange for nutrients like nitrogen and phosphorus that it can access more easily than the plants can. The fungus is also pursuing its own agenda, growing toward resources, forming connections that benefit its own survival and reproduction. And somehow, in the process of all this activity, nutrients and chemical compounds end up moving between plants in ways that can affect their growth and survival. Perhaps the most famous type of mycorrhizal connection is the ectomycorrhizal relationship, common in temperate and boreal forests. In these associations, the fungal hyphae form a sheath around the outside of fine root tips and grow between the cells of the root tissue, creating an enormous surface area for exchange without actually penetrating into the root cells. This dramatically increases the root's ability to absorb water and nutrients, effectively expanding the root system of the tree by orders of magnitude. The tree, in turn, provides the fungus with sugars, supporting the growth of the mycelial network through the soil. These ectomycorrhizal fungi include many of the mushrooms we're most familiar with, the species we see fruiting on the forest floor in autumn. Boletes with their sponge-like pores under their caps. Rasulas with their bright colors and brittle flesh. Amanitas, including both edible species and deadly poisonous ones. All of these are part of this hidden network, their fruiting bodies emerging briefly above ground while the real organism, the vast mycelium, remains hidden below, doing its slow work of connecting and nourishing. The other major type of mycorrhizal relationship is arbuscular mycorrhizae, which forms in the majority of plant species worldwide, including most crop plants, grasses, and tropical trees. In this type of association, the fungal hyphae actually penetrate into the root cells, forming highly branched structures called arbuscules that look like tiny trees within the cell. These structures are where most of the exchange happens, nutrients flowing from fungus to plant and sugars flowing from plant to fungus, all mediated by specialized membrane systems that keep the two organisms separate while allowing transfer between them. Arbuscular mycorrhizal fungi are ancient, their lineage stretching back 400 million years or more, and they're found in almost every terrestrial ecosystem. Unlike ectomycorrhizal fungi, which produce visible mushrooms, arbuscular mycorrhizal fungi produce microscopic spores in the soil, invisible to the naked eye but present in enormous numbers. These fungi have co-evolved with plants for so long that many plants simply cannot grow well without them. The fungi have become essential partners, as fundamental to the plant's survival as its own roots. The idea that these fungal networks might facilitate communication between trees captured public imagination when it was first proposed. The possibility that trees might be warning each other of insect attacks, or that older mother trees might be nurturing their offspring through fungal connections, resonated with something deep in our understanding of nature. It suggested that forests are not battlefields of competition but rather communities of cooperation, that trees are not isolated individuals but members of a vast, interconnected society. Scientists continue to debate exactly how much of this popular narrative is supported by solid evidence. Some researchers caution that while transfer of substances between plants through fungal networks has been demonstrated, it's not always clear whether this transfer is significant enough to affect plant fitness, or whether it represents true communication versus simply a side effect of the fungus pursuing its own interests. The fungus, after all, is not a passive cable. It's a living organism with its own needs and strategies. What we can say with certainty is that fungal networks are real, vast, and important. They connect plants in physical ways that can affect the movement of water, nutrients, and chemical compounds. They create a more integrated ecosystem where the health and success of one plant can influence its neighbors through shared fungal partners. They represent a layer of interaction and interdependence that we're only beginning to fully understand. The sheer scale of some individual fungal networks is staggering. The largest known organism on Earth is a honey fungus, Armillaria astoi, growing in Oregon's Malheur National Forest. This single fungal individual covers approximately 890 hectares, roughly 3.4 square miles, and is estimated to be over 2,000 years old. Most of its mass is underground mycelium, spreading through the soil, growing slowly but persistently, exploring its environment, forming connections, breaking down dead wood, producing the occasional mushroom cluster when conditions are right. Imagine this organism, this vast living network, existing largely unnoticed, going about its business for centuries while empires rose and fell, while humans invented agriculture and built civilizations, while forests grew and were cut down and grew again. It represents a kind of time scale and spatial scale that's completely different from our human experience, a reminder that there are other ways of being, other patterns of existence, playing out all around us if we only know how to look for them. These networks aren't static. They're constantly growing, changing, responding to conditions. Hyphae extend into new territory, exploring, seeking resources. Existing connections can strengthen or weaken depending on the flow of nutrients. Sections of mycelium can die back if resources become scarce, while other sections flourish when conditions are good. The network is alive, dynamic, constantly adjusting to the shifting landscape of opportunity in the soil. For the plants connected to these networks, the benefits can be substantial. Studies have shown that mycorrhizal plants often grow better than non-mycorrhizal plants, especially in challenging conditions. The fungal network provides access to nutrients that would otherwise be beyond the plant's reach, helps the plant withstand drought by improving water uptake, and may even provide some protection against soil pathogens. In return, the plant supports a vast underground network that extends far beyond its own root system, a network that connects it to its neighbors and to the larger forest community. There's something peaceful in contemplating these networks, these slow-growing, persistent connections spreading through the soil. They remind us that there are patterns of relationship and interdependence that operate on different timescales than our daily lives. They exist in the quiet spaces, doing their work unseen, asking nothing of us, simply being part of the great cycles of the forest. And knowing that they're there, that beneath every step we take in the forest there's this vast and intricate network of life, somehow makes the world feel richer, more connected, more deeply alive. The fascinating biology of mushrooms. The life of a mushroom begins in a way that might seem almost magical if we didn't understand the biology behind it. Hidden in the soil or within decaying wood, A network of fungal mycelium grows slowly, steadily, year after year, breaking down organic matter, absorbing nutrients, forming connections with roots. For months or even years, this mycelium exists in a vegetative state, growing but not reproducing, simply doing the quiet work of being a fungus. And then, when conditions are just right, when temperature and moisture and other factors align in the perfect way, something remarkable happens. The mycelium begins to produce a mushroom. The mushroom itself is a fruiting body, a specialized reproductive structure whose sole purpose is to produce and disperse spores. The real organism, the living being that persists through time, is the mycelium. The mushroom is temporary, ephemeral, a brief eruption into the visible world before withering away once its job is done. And yet, what a remarkable structure it is. The process begins with tiny knots of hyphae forming in the mycelium, dense aggregations of fungal threads that will become the mushroom. These knots, called primordia, can remain dormant for long periods, waiting for the right conditions. When rain comes after a dry spell, or when temperatures shift to the right range, or when some other environmental trigger occurs, these primordia begin to grow rapidly. Cells divide, absorb water, expand, and in a matter of days or sometimes mere hours, a mushroom pushes up through the soil or emerges from a dead log. The speed of mushroom growth is remarkable. A mushroom doesn't grow like a tree, slowly adding new tissue to existing structure. Instead, the cells of the mushroom are already present in the primordium, just waiting to expand. When water becomes available, these cells absorb it rapidly, swelling like tiny balloons, and the mushroom literally inflates into its final form. This is why mushrooms seem to appear overnight. Why a forest floor that was bare yesterday can be dotted with mushrooms after a night of rain. They were always there, just waiting, compressed and dormant beneath the surface, ready to emerge when the moment was right. Once a mushroom has expanded to its full size, it begins the real work of producing spores. The location of spore production varies depending on the type of mushroom. In gilled mushrooms, the kind we most commonly recognize with a cap and stem and radiating gills underneath, the spores form on the surfaces of these gills. In boletes and polypores, they form in tiny tubes that create a pore-like surface. In other mushrooms, they might form on the surfaces of upward-pointing branches or inside enclosed chambers. But regardless of the specifics, the principle is the same. Maximize surface area for spore production and create a mechanism for releasing those spores into the air. A single mushroom can produce an astonishing number of spores. Estimates vary, but a large mushroom might release billions of spores over the course of its brief life. Tiny microscopic packages of genetic information, each one capable of starting a new fungal colony if it lands in the right conditions. Most of these spores will not succeed. They'll land in unsuitable locations or fail to find the resources they need to germinate, or be eaten by soil microorganisms. But by producing spores in such vast numbers, the fungus ensures that at least a few will find their way to favorable conditions and establish new colonies. The spores themselves are marvels of natural engineering. Each one is microscopic, typically only a few micrometers across, small enough to be carried for miles on wind currents. They're surrounded by a tough wall that protects them from desiccation, UV radiation, and other environmental stresses. Some spores can remain viable for years, waiting patiently in the soil for conditions that will trigger germination. Different species produce spores in different colors: white, cream, pink, brown, purple, black, creating the spore prints that mushroom enthusiasts use to help identify species. When a spore lands in a suitable location and conditions are right, it germinates, sending out a thin hypha that begins to grow through its substrate. But here's where things get interesting. In most mushroom-forming fungi, this single hypha cannot, by itself, form a complete fungal colony. It needs to encounter another compatible hypha from a different spore. When two compatible hyphae meet, they fuse, combining their genetic material, and from this fusion, a new mycelial network can develop, one that has the genetic diversity to eventually produce mushrooms of its own. This means that the mycelium growing in the forest, the network that will eventually produce mushrooms, is actually the result of a successful mating between two separate hyphae that originated from different spores. In a sense, the mycelium is the individual, the living organism, and the mushrooms it produces are its reproductive structures, analogous to flowers on a plant. But unlike most organisms we're familiar with, the main body of the fungus is hidden, living underground or within its substrate, and the only part we typically see is the reproductive structure, the mushroom itself. Some mushrooms have evolved fascinating specialized structures for spore dispersal. Puffballs, those round fungi that can be kicked to release clouds of spores, accumulate millions of spores inside their hollow interior, then develop a hole at the top through which the spores can escape when the puffball is disturbed by rain, wind, or a passing animal. Stinkhorns produce foul-smelling slime containing their spores, attracting flies that then carry the spores away on their bodies. Birds' nest fungi create tiny cup-shaped structures filled with spore-containing packets that splash out when raindrops hit them, effectively using raindrops as a launching mechanism. Perhaps most mysteriously, some mushrooms produce light. These bioluminescent fungi glow in the dark, emitting a soft greenish light at a wavelength of around 520 to 530 nanometers. More than 125 species of bioluminescent fungi are known, found largely in temperate and tropical climates, all of them sharing the same basic biochemical mechanism for light production. They contain a compound called luciferin that, when combined with oxygen and specific enzymes, releases light as a byproduct of the chemical reaction. Why would a mushroom glow? For years, this was a mystery, but recent research suggests that the light attracts insects that help disperse spores. Experiments have shown that glowing mushrooms attract more arthropods than non-glowing mushrooms, particularly in the dark understory of closed canopy forests where other forms of spore dispersal might be less effective. The mushrooms glow continuously, day and night, but their glow is only visible in darkness, creating an eerie, magical sight in nighttime forests where these species grow. The bioluminescence can occur in different parts of the mushroom depending on the species. In some, it's the gills that glow. In others, it's the cap or the stem or even just the mycelium in the substrate. The intensity varies too. Some species produce only a faint glow barely visible to dark-adapted eyes. Others, like some species found in Brazil and Southeast Asia, can glow brightly enough to read by. Indigenous peoples in some regions have traditionally used glowing mushrooms as natural flashlights, carrying them to light their way through dark forests. The fact that bioluminescence evolved independently in fungi at least 3 or 4 times in different lineages suggests that it provides a real advantage. It's not just a random byproduct of metabolism, but an adaptation that helps these fungi reproduce. The light is controlled by a circadian clock, meaning the fungi can regulate when they glow most intensely, typically increasing light production at night when it's most likely to attract insect attention. This level of control implies sophisticated biological machinery working behind the scenes, the fungus responding to day-night cycles, regulating its biochemistry to maximize the effectiveness of its light production. Mushrooms have also evolved remarkable abilities to survive in extreme conditions. Some species can withstand freezing temperatures, their cells protected by compounds that prevent ice crystals from forming and damaging delicate cellular structures. Others can survive in extremely dry conditions, drying out completely and then reviving when water becomes available again. Fungal spores are even more resilient, with some species producing spores that can survive in space, exposed to vacuum, radiation, and temperature extremes, a testament to the robust nature of fungal biology. The pigments that give mushrooms their incredible diversity of colors serve various purposes. Some are simply byproducts of the compounds the mushroom produces, giving it color as an incidental effect. Others actively protect the mushroom from UV radiation, important for species that fruit in exposed locations. Still others may serve as warning colors, signaling to potential predators that the mushroom is toxic or unpalatable. The bright red of the fly agaric mushroom with its white spots, one of the most iconic mushroom images, is thought to serve this purpose, a visual advertisement that eating this mushroom would be a bad idea. Some mushrooms can even respond to mechanical stimulation, changing color when touched or damaged. The species of Lactarius bleed latex when cut, sometimes white, sometimes colored, sometimes changing color on exposure to air. Boletes often bruise blue or green when handled, the result of rapid oxidation of compounds in their flesh. These color changes are chemical reactions happening in real time, the mushroom's tissues responding to damage, producing compounds that may help prevent infection or discourage further damage from whatever caused the initial injury. The textures of mushrooms are equally diverse. Some are slimy, covered in a thick layer of mucus that may help prevent desiccation or deter predators. Others are dry, velvety, or covered in tiny scales or hairs. Some are tough and woody, capable of persisting for years on a tree trunk. Others are soft and fragile, lasting only a day or two before collapsing into a puddle of decaying tissue. This diversity reflects the enormous range of strategies fungi have evolved for surviving and reproducing in different environments. And then there are the truly unusual mushrooms, the ones that challenge our expectations of what a mushroom should look like. Coral fungi that branch like underwater corals, creating intricate structures that could be mistaken for something from the ocean. Cup fungi that form tiny bowls or goblets, sometimes brilliantly colored, holding a reservoir of spores waiting to be splashed out by rain. Earth stars that begin as round balls but then split open, the outer layer peeling back in pointed segments to create a star shape surrounding the inner spore sac. Each of these represents a different solution to the same basic challenge: how to produce and disperse spores as effectively as possible. The life cycle of a mushroom, from spore to mycelium to mushroom to spore again, is a cycle that has been repeating for hundreds of millions of years, a pattern so successful that it has persisted through mass extinctions, through dramatic changes in climate and vegetation, through the rise and fall of countless other species. And it continues today in forests and fields around the world, these quiet organisms doing their work of reproduction, spreading their spores, establishing new colonies, connecting with plant roots, breaking down organic matter, existing in their patient, persistent way. There's something deeply peaceful about understanding this cycle, about knowing that mushrooms aren't trying to do anything except continue their species, that they're not concerned with our human dramas or worried about our problems. They simply are, growing when conditions permit, producing their spores, then fading away, the mycelium returning to its quiet work in the soil. This has been happening since long before humans existed, and it will continue long after we're gone, a cycle of such fundamental importance to the functioning of ecosystems that without it, the world as we know it could not exist. Mushrooms in ecosystems, in the grand cycles of nature, in the constant turning over of matter from living to dead to living again, mushrooms play a role that cannot be overstated. They are the planet's primary decomposers, the organisms responsible for breaking down the vast majority of dead plant material on land, recycling nutrients that would otherwise remain locked away in dead tissues, making those nutrients available once again for new life to use. Without mushrooms and their fungal relatives, the world would look drastically different. Dead plant material would accumulate endlessly. The carbon that plants capture from the atmosphere through photosynthesis would remain trapped in their tissues. The cycling of nutrients would slow to a crawl. Ecosystems as we know them would cease to function. Consider a fallen tree in a forest. It might weigh several tons, all of that weight built from carbon, nitrogen, phosphorus, and other elements that the tree extracted from air, water, and soil over decades or centuries of growth. When the tree falls, either from wind or disease or old age, it represents an enormous resource, a mother lode of nutrients waiting to be released. But trees are not easy to break down. Their wood is composed primarily of cellulose and lignin, complex compounds that are remarkably resistant to decomposition. Most organisms cannot digest these materials at all. Fungi, however, have evolved the enzymatic machinery necessary to break down even the most recalcitrant plant materials. They produce a whole suite of enzymes that can work on different components of wood and plant tissue. Some enzymes attack cellulose, breaking it down into simpler sugars. Others work on lignin, the extremely tough polymer that gives wood its strength and makes it so resistant to decay. Different fungi specialize in different strategies. White rot fungi can break down both cellulose and lignin, leaving behind pale, soft, spongy wood. Brown rot fungi preferentially break down cellulose, leaving behind brown, fragmented wood that crumbles when touched. The process of decomposition is slow, measured in years or decades for large pieces of wood. The mycelium of decomposer fungi grows through the wood, following the grain, extending into every crack and crevice, secreting enzymes that break down the wood into smaller molecules that the fungus can absorb. As the fungus grows and feeds, it doesn't simply consume the wood. It transforms it, breaking complex molecules into simpler ones, releasing nutrients into the surrounding environment, creating habitat and resources for countless other organisms. A dead log lying on the forest floor becomes, over time, a hotbed of biological activity. The fungal mycelium permeating the wood provides food for insects, mites, and other invertebrates. These creatures tunnel through the softening wood, fragmenting it further, creating more surface area for fungi to colonize. Their droppings, containing partially digested wood and fungal tissue, enrich the soil around the log. Salamanders and small mammals use the log for shelter. Mosses and ferns grow on its surface. The log becomes less a single dead object and more a complex ecosystem in miniature, all of it made possible by the slow, patient work of decomposer fungi. And eventually, perhaps after decades, the log will be completely incorporated back into the soil. The nutrients it contained will have been released, made available for new plants to use. The carbon in its wood will have been partly released as carbon dioxide, returning to the atmosphere where plants can capture it again through photosynthesis, and partly incorporated into the bodies of the countless organisms that fed on the decaying wood. The log will have disappeared, but nothing will have been lost. Everything will have been transformed, recycled, made part of the great cycle of growth and decay that sustains all life. This process plays out everywhere that plants grow and die, which is to say, everywhere on land. In forests, grasslands, wetlands, even in your backyard, fungi are constantly at work breaking down dead plant material. They decompose fallen leaves, dead roots, grass clippings, flower stems, everything that plants produce and then shed or that dies when the plant dies. The speed of decomposition varies depending on climate, moisture, temperature, and the type of material being decomposed, but the principle remains the same. Fungi are doing the essential work of recycling. In tropical rainforests, where warmth and moisture are abundant, decomposition happens quickly. A fallen leaf might be completely broken down in a matter of weeks or months. In colder regions or in areas with seasonal droughts, the process is slower, but it's still happening, the fungi working at whatever pace the conditions allow. In the far north, in boreal forests where cold temperatures slow all biological processes, dead wood can persist for decades, colonized by successive waves of different fungal species, each one specialized for breaking down wood at different stages of decay. The mushrooms we see fruiting on dead wood or leaf litter are the visible signs of this hidden work. They are the reproductive structures of these decomposer fungi, emerging to release spores that will colonize new dead material. Some of them are quite beautiful. The delicate fairy inkcap mushrooms that grow on wood chips, the bright orange chicken of the woods that appears on dead or dying trees, the elegant oyster mushrooms that grow in overlapping shelves on hardwood logs. Each mushroom represents a fungus busy at work recycling the dead material it's growing on. But fungi don't just decompose plant material. Some species are predators, capturing and consuming small animals, usually nematodes or other microscopic soil organisms. These predatory fungi produce specialized structures, tiny loops or branches that can trap their prey. When a nematode swims or crawls through the soil and encounters one of these traps, it triggers a response. In some species, the trap inflates rapidly, constricting around the nematode and holding it fast. In others, the trap is coated with a sticky substance that glues the nematode in place. Once captured, the fungus grows hyphae into the nematode's body, digesting it from the inside, absorbing the nutrients it contains. It's a remarkable adaptation, allowing fungi to supplement their diet with nitrogen and other nutrients obtained from animal prey. Other fungi are parasites, feeding on living plants, animals, or other fungi. Some plant parasitic fungi cause diseases that can kill their hosts, while others establish chronic infections that weaken but don't immediately kill the plant. Certain fungi parasitize insects, with some species capable of manipulating their host's behavior. The famous zombie ant fungi, for instance, infect ants and alter their behavior, causing them to climb to elevated locations before they die, positions from which the fungus can more effectively disperse its spores. It's a disturbing example of how sophisticated fungal strategies can be, how they've evolved to exploit every available ecological opportunity. But most fungi in healthy ecosystems are either decomposers or mutualists, living in beneficial relationships with plants. These fungi are fundamental to nutrient cycling. When they break down dead organic matter, they release nutrients in forms that plants can use. The nitrogen that was locked up in dead leaves becomes available again. The phosphorus that was part of old root systems returns to the soil solution where new roots can absorb it. The fungal mycelium acts as a living recycling system, constantly turning over nutrients, keeping them moving through the ecosystem. This role is especially critical in nutrient-poor environments. In tropical rainforests, for instance, most of the nutrients in the ecosystem are held in living biomass rather than in the soil. The soils themselves are often ancient, heavily weathered, and nutrient depleted. But decomposition is so rapid and efficient, and mycorrhizal fungi are so effective at capturing released nutrients and delivering them to plant roots, that these forests sustain incredible biodiversity and productivity despite their poor soils. The fungi are the key, the living link that keeps nutrients cycling rapidly from dead tissue to fungus to plant and back again. In temperate forests, the seasonal pattern of leaf fall creates pulses of organic material that fungi work through over the course of the year. In autumn, when leaves fall, the forest floor becomes carpeted with fresh litter. Through the winter, this litter begins to break down, slowly, as cold temperatures limit fungal activity. But when spring comes and temperatures rise, fungal growth accelerates. The mycelium in the leaf litter becomes active again, growing rapidly, breaking down the accumulated organic matter. By summer, much of the previous autumn's leaf fall has been substantially decomposed, its nutrients released and captured by new plant growth. Different fungal species specialize in breaking down different materials and appear in predictable sequences. Some species are the first to colonize fresh litter, quickly breaking down the most accessible materials. Other species arrive later, working on the more resistant compounds that early colonizers can't metabolize. Still others appear only near the end of the decomposition process, when the original material has been transformed almost beyond recognition. This succession of fungal species, each playing its part in the complete decomposition of organic matter, is one of the hidden patterns of the forest, a choreography of decay that plays out constantly beneath the visible world. Fungi also play crucial roles in carbon cycling at larger scales. They influence how much carbon from dead plant material is released back to the atmosphere as carbon dioxide versus how much becomes stabilized in soil organic matter. Some fungal species produce compounds that resist further decomposition, effectively sequestering carbon in the soil for long periods. Others release most of their captured carbon quickly as carbon dioxide. The balance between these different fungal strategies, along with factors like climate and plant community composition, determines how much carbon forests and other ecosystems can store long-term. Recent research has shown that mycorrhizal fungi can store significant amounts of carbon in their mycelial networks, essentially locking it away underground in living fungal tissue. A single large tree can be connected to mycorrhizal fungi whose combined mycelium weighs hundreds of pounds, all of that fungal tissue built from carbon compounds provided by the tree. When the tree dies and stops supplying sugars to the fungi, much of that fungal mycelium dies too, adding organic matter to the soil. Some of this carbon will be quickly respired back to the atmosphere, but some will be incorporated into stable soil organic matter, representing carbon that's been effectively removed from the atmosphere for decades or longer. The relationship between fungi and climate regulation goes even deeper. By facilitating plant growth through mycorrhizal partnerships, fungi enable plants to capture more carbon dioxide from the atmosphere. By breaking down dead plant material, they release carbon back to the atmosphere but also create soil conditions that allow new plants to grow. They're part of the vast, interconnected system that regulates the flow of carbon between atmosphere, living things, and soil. A system so complex and so important that understanding it better is one of the major challenges of contemporary ecology. Mushrooms also provide food for an enormous variety of animals. Slugs and snails consume mushrooms, as do many insects. Squirrels harvest and cache mushrooms for winter food. Deer browse on mushrooms when they're abundant. Even large mammals like bears will eat mushrooms when they're available. Some animals are specialists, depending heavily on mushrooms as a food source. Flying squirrels in North American forests eat primarily truffles, the underground fruiting bodies of certain mycorrhizal fungi. They have an acute sense of smell that allows them to locate truffles beneath the soil surface, and their digestive systems are specialized for processing fungal tissue. When these animals eat mushrooms, they serve the fungus by dispersing spores. Spores that pass through an animal's digestive system often remain viable and can be deposited in new locations along with a convenient package of fertilizer in the form of the animal's droppings. This creates a mutualism. The animal gets food, and the fungus gets spore dispersal to new areas. Some fungi have evolved to smell attractive to specific animals, producing compounds that mimic the scents these animals are naturally drawn to, effectively advertising their presence and encouraging consumption. In this way, fungi are woven into food webs at multiple levels. They're primary decomposers, breaking down dead material. They're producers of mushroom tissue that feeds animals. They're partners with plants, helping them grow and therefore supporting all the animals that depend on those plants. They're predators of microscopic soil animals. They're themselves consumed by specialized organisms that feed on fungal tissue. They're everywhere in the ecosystem, playing multiple roles simultaneously, connecting different parts of the food web in ways that aren't always obvious but are always important. The seasonal patterns of mushroom fruiting create temporal rhythms in ecosystems. In temperate regions, mushroom season typically comes in autumn, when cooling temperatures and increased rainfall create ideal conditions for fruiting. The forest floor that was relatively bare of mushrooms in summer suddenly explodes with them, species after species emerging, often in characteristic sequences, different species appearing at different times as the season progresses and conditions change. This pulse of mushroom availability provides a seasonal resource for animals, a bounty that appears reliably each year and that many animals have learned to time their own activities around. In tropical regions, patterns are different, often tied to rainy seasons rather than temperature changes. Monsoon forests might have 2 peak fruiting periods per year, each following seasonal rains. Rainforests with less pronounced seasonality might have mushrooms fruiting year-round, though even there, certain species appear only at certain times or only under particular conditions. These patterns reflect the deep integration of fungi into their ecosystems. Their responses to environmental conditions shaped by millions of years of evolution in specific places with specific seasonal rhythms. Even in the quietest moments, when no mushrooms are visible and the forest seems still, the fungi are at work. The mycelial networks in the soil are growing, exploring, connecting with roots, breaking down organic matter. The decomposition of that dead log continues. The nutrient cycling happens. The partnerships between fungi and plant roots maintain themselves, trading sugars for nutrients, keeping both partners alive and healthy. The ecosystem continues to function because fungi continue their quiet, essential work. Understanding this role, this essential position that fungi occupy in every terrestrial ecosystem, changes how we see forests and fields. They're not just collections of plants with animals living among them. They're complex networks where fungi form the living links between components, where the hidden underground world of mycelium is just as important as the visible world of trees and flowers. The mushrooms we see are beautiful and fascinating in their own right, but they're also reminders of all this hidden activity, signs pointing to the vast and intricate workings of the ecosystem happening all around us, mostly out of sight. The diversity of mushroom forms. If you were to catalog all the different shapes, colors, textures, and sizes of mushrooms, you would find yourself faced with a diversity that rivals flowers in its variety and surpasses them in its strangeness. Mushrooms are not constrained by the evolutionary pressures that have shaped plants toward certain recognizable forms. They've taken countless evolutionary pathways, each species solving the problem of spore production and dispersal in its own unique way, resulting in forms that range from the familiar to the bizarre, from the beautiful to the grotesque, from the tiny to the enormous. Let's start with size because the range is remarkable. At the small end of the spectrum are mushrooms so tiny they're easy to overlook entirely. Species like the tiny pin mushrooms that fruit on dung or dead leaves might have caps only 1 millimeter or 2 across, stems like fine threads, the whole mushroom so delicate it could fit on the tip of your smallest finger with room to spare. These miniature mushrooms often live their entire lives in just a few days, appearing, maturing, releasing spores, and withering away while larger mushrooms are still slowly expanding. At the other extreme are the giants of the mushroom world. The largest fruiting body ever reliably documented was a specimen of the chicken of the woods mushroom weighing approximately 100 pounds. Artist's conch, a woody polypore that grows on tree trunks, can produce fruiting bodies several feet across that persist for years, adding new layers of tissue each growing season, building up structures that can weigh 50 pounds or more. And then there's the underground giant, the largest organism on Earth, that Armillaria fungus in Oregon whose mycelial network spans square miles, though its individual mushrooms are quite modest in size. But it's not just overall size that varies. Consider the shapes mushrooms can take. The classic toadstool form, with its cap and stem and gills underneath, is just one possibility among many. Some mushrooms have caps but no stems, growing directly from their substrate in overlapping shelves or brackets. Others have stems but no distinct caps, instead branching like corals or forming clubs. Still others are spherical or cup-shaped, or form irregular masses, or look like elongated fingers reaching up from the ground. Gilled mushrooms, the most recognizable type, have evolved their structure in response to a particular challenge: How to maximize the surface area available for producing spores while protecting those spore-producing surfaces from rain and debris. The solution is elegant. A cap held up by a stem, with the spore-producing tissue arranged in thin, radiating plates underneath the cap. Gravity pulls the spores downward when they're released, allowing them to fall free of the gills and catch air currents for dispersal. The cap protects the gills from direct rainfall while allowing air circulation. It's a design so successful that it's evolved independently in multiple different lineages of fungi. But the gilled form has countless variations. Gills can be closely spaced or widely spaced, attached to the stem in different ways or completely free from it, thick or thin, waxy or brittle, forked or simple. Caps can be flat, convex, conical, bell-shaped, funnel-shaped, or irregular. They can be smooth, hairy, scaly, slimy, or covered in warts or patches. Stems can be thick or thin, short or tall, equal in width throughout or bulbous at the base, hollow or solid, smooth or covered in scales or rings. Colors are equally diverse. Mushrooms come in essentially every color you can imagine, and some you probably can't. White is common, as are various shades of brown, tan, and gray, subtle colors that blend with leaf litter and tree bark. But there are also brilliant reds, like the classic fly agaric with its scarlet cap and white spots. Deep purples, like the Cordyceps species whose caps can be almost violet. Bright yellows and oranges, like the golden chanterelles that glow like flames in the forest understory. Greens are rarer but exist, like the parrot mushroom, one of the most spectacularly colored species. Some mushrooms combine multiple colors in striking patterns. The cap might be one color while the gills are another and the stem a third. Colors might form zones or rings on the cap, or radiate outward in rays, or appear as spots or patches. As mushrooms age, their colors often change. A young mushroom might be brightly colored, then fade as it matures or start pale and darken, or change to completely different colors, sometimes as a response to environmental conditions like moisture or light. Textures are just as variable as colors and shapes. Some mushrooms are slimy, their caps covered in a thick layer of mucilage that makes them glistening and slippery. Others are dry and velvety, pleasant to touch. Some are warty or scaly, their surfaces broken up into distinct patches or scales. Many are smooth and silky. Some feel waxy, some cottony, some tough and leathery. The chanterelle, prized by mushroom foragers, has a distinctively smooth, almost rubbery texture that's part of how experienced hunters identify it. The flesh of mushrooms varies too. Some species have firm, dense flesh that holds its shape when sliced. Others are fragile and brittle, shattering when handled. Some have flesh that exudes latex when cut, droplets of liquid that can be white, colored, or even changing color on exposure to air. The milk cap mushrooms in the genus Lactarius are named for this characteristic. When you cut a milk cap, it bleeds, sometimes copiously, the latex flowing from the damaged tissue in a way that can seem almost animal-like. Then there are the mushrooms that don't follow the standard cap and stem format at all. Puffballs are spherical, ranging in size from marble size to larger than a basketball. Young puffballs have solid white flesh inside, but as they mature, this flesh transforms into billions of spores, and the interior becomes filled with a powder of spores that puffs out through an opening at the top when the puffball is disturbed. Earth stars are similar but split open in a star pattern, the outer layer peeling back to reveal the inner spore sac. Stinkhorns are among the strangest mushrooms. They start as egg-shaped structures underground or in leaf litter. When mature, the mushroom rapidly expands, breaking out of the egg and elongating into bizarre shapes. Some stinkhorns look like fingers or tentacles. Others resemble, rather unmistakably, certain parts of human anatomy, complete with coloration that enhances the resemblance. The similarity is so striking that one common species is called the devil's dipstick, and another, found in East Asia, is sometimes called bamboo mushrooms or bamboo pith. The caps of stinkhorns are covered in a foul-smelling slime containing spores. This slime smells like rotting meat or feces, attracting flies that then disperse the spores. Coral fungi grow in branching, upright clusters that really do look like coral, some forming compact tufts only inches high, others creating intricate structures over a foot tall. They can be white, yellow, orange, pink, or purple, and they fruit directly from the soil or from decaying wood, their branches reaching upward like tiny frozen explosions. The spores form on the surface of the branches, all over the structure, rather than being concentrated on a single specific surface. Cup fungi create bowl-shaped or cup-shaped fruiting bodies, ranging in size from tiny cups a few millimeters across to large, saucer-sized structures. The spores form on the inner surface of the cup. Many cup fungi are brightly colored, brilliant orange or red or yellow, creating surprising splashes of color against the browns and greens of the forest floor. Some cup fungi have unusual shapes, like the violet-crowned cup fungus, which has a series of crown-like points around its rim, or the devil's urn, a dark, urn-shaped fungus that fruits in early spring. Bird's nest fungi are small, typically less than half an inch tall, but they're remarkable in their engineering. They form tiny nest-shaped cups containing what look like eggs. These eggs are actually packets of spores called peridioles. When a raindrop hits the nest, it splashes the peridioles out, potentially launching them several feet away. The peridioles are sticky and can adhere to vegetation, where they'll later release their spores. It's a remarkable adaptation, using the force of raindrops as a launching mechanism, and it works well enough that bird's nest fungi are found worldwide. Jelly fungi have a gelatinous texture, often translucent or semi-translucent, and they can dry out completely in dry weather, then rehydrate when rain comes. The most familiar is probably the wood ear mushroom, used in Asian cuisine, which grows on elder trees and has a distinctively ear-like shape and a soft, gelatinous texture when fresh. Witch's butter is another jelly fungus, bright orange or yellow, growing in gelatinous masses on dead wood, especially after rain. Some mushrooms are tough and woody, persisting for years rather than days. Polypores, also called bracket fungi or shelf fungi, grow on trees, living or dead, producing fruiting bodies that can last for years, adding new layers of spore-producing tissue each growing season. The undersides of these mushrooms are covered in tiny pores, hence the name, each pore the opening of a tube where spores are produced. Some polypores can become enormous, weighing many pounds, projecting from tree trunks like wooden shelves. The artist's conch gets its name from the fact that its white pore surface bruises brown when scratched, allowing artists to create detailed drawings on the surface of the fresh mushroom. which then persist as the mushroom dries and hardens. These decorated mushrooms become permanent artworks, the fungal tissue preserving the image indefinitely once dried. Tooth fungi hang downward pointing spines or teeth from the undersurface of the cap, rather than gills or pores. Spores form on the surface of these teeth and drop free when mature. Some tooth fungi are quite beautiful, like the lion's mane mushroom, which produces cascading masses of long, white spines that can make it look like a white waterfall or icicle hanging from a tree. Others are less conspicuous but equally fascinating in structure. Chanterelles have a vase-shaped or trumpet-shaped form, often with a wrinkled or ridged undersurface rather than true gills. These ridges are blunt and forked, running down the stem, and they're one of the key features that distinguish chanterelles from potentially toxic lookalikes. Chanterelles are prized edible mushrooms, and part of their appeal is their distinctive form. Their bright colors, usually golden yellow or orange, and their fruity fragrance, often described as reminiscent of apricots. Morels are among the most prized edible mushrooms, and they're distinctive in appearance, with caps that are covered in pits and ridges creating a honeycomb or sponge-like texture. They're hollow inside, both cap and stem, a feature that helps distinguish them from false morels, which can be toxic. True morels fruit in spring, often in association with certain trees or in areas that have been disturbed by fire, and mushroom hunters wait eagerly for morel season each year. Truffles are perhaps the most unusual mushrooms, at least in terms of lifestyle. They're hypogynous, meaning they fruit underground, never emerging above the soil surface. They're typically round or irregular in shape, ranging from marble-sized to as large as a fist. Their exterior is warty or smooth, and their interior is marbled with veins of spore tissue and sterile tissue. Because they're underground, they can't release spores into the air. Instead, they've evolved to be found and eaten by animals, relying on animals to disperse their spores in their droppings. Truffles have evolved strong, distinctive odors to attract animals. These odors can be earthy, garlicky, or even chemical smelling, depending on the species. Some species, particularly European species in the genus Tuber, produce odors that humans find extremely appealing, and these truffles are among the most expensive foods in the world, hunted with specially trained dogs or pigs and selling for hundreds or thousands of dollars per pound. The diversity doesn't stop with form. Consider the different reproductive strategies. Some mushrooms fruit gregariously, dozens or hundreds of individuals appearing close together from a shared mycelial network. Others fruit solitarily, individual mushrooms spaced well apart. Some fruit in rings, the famous fairy rings, where the expanding mycelial network fruits at its outer edge, creating a ring of mushrooms that expands outward year by year, sometimes persisting for decades or even centuries in the same location. Some mushrooms are ephemeral, appearing and disappearing in a day. The shaggy mane mushroom, for instance, autodigests, its gills liquefying from the bottom up after the spores mature, literally dissolving itself to help disperse its spores, leaving behind nothing but a black puddle and a stem by the end of the day. Others persist for weeks, slowly releasing spores day after day. Still others, the woody polypores, persist for years, fruiting once and then maintaining that fruiting body year after year, producing new spores each growing season from the same structure. All of this diversity, all these different forms and colors and textures and strategies, represents millions of years of evolution, fungi adapting to specific niches, solving the challenges of their environments in countless different ways. Each mushroom form is a solution to a problem, a way of producing and dispersing spores that works in particular conditions for particular species in particular ecosystems. And when you walk through a forest and see this diversity firsthand, when you see 10 different mushroom species, each with its own distinctive appearance, you're seeing evolution at work, the result of countless generations of fungi adapting, changing, exploring the possible space of fungal form. Mushrooms and human history. Humans and mushrooms have a relationship that stretches back into the mists of prehistory, back to a time before writing, before agriculture, when our ancestors lived as hunter-gatherers, intimately connected to the landscapes they inhabited and keenly aware of the seasonal rhythms of the organisms around them. Mushrooms would have been part of their world, emerging after rains, providing food, perhaps serving other purposes we can only guess at now. Though we can't know for certain how our earliest ancestors interacted with mushrooms, we can see evidence of mushroom use throughout recorded history and infer that the relationship is ancient. One of the earliest pieces of archaeological evidence for human use of mushrooms comes from the preserved body of Ötzi the Iceman, a man who died in the Alps approximately 5,300 years ago. When his remarkably well-preserved remains were discovered in 1991, researchers found that he was carrying two types of mushrooms. One was amadou, a bracket fungus that burns slowly when dried, useful for transporting fire. The other was birch polypore, strung on a leather cord, possibly used for its medicinal properties, as this fungus has been shown to contain compounds with antibiotic and antiparasitic effects. This single discovery tells us that by the Copper Age, people in Europe understood mushrooms well enough to identify useful species, knew how to prepare and carry them, and incorporated them into their daily lives. Ötzi wasn't carrying these mushrooms by accident. He selected them deliberately, carried them with him as valuable tools, suggesting a depth of traditional knowledge about fungi that must have been passed down through generations. In ancient China, mushrooms held an especially important place in traditional medicine. The reishi mushroom, known in Chinese as lingji, has been used medicinally for at least 2,000 years. Ancient texts refer to it as the mushroom of immortality, and it was reserved for emperors and nobility, so valued that it appears in countless works of Chinese art. Its distinctive kidney-shaped cap with a varnished, reddish-brown surface rendered in paintings and carvings. Traditional Chinese medicine attributed various healthful properties to reishi, and modern research has found that it does contain bioactive compounds with potential medicinal effects, though its actual medical benefits are still being studied. Other mushrooms featured prominently in traditional Chinese medicine as well. Shiitake mushrooms were cultivated in China possibly as early as the 12th century, making them one of the first mushrooms to be deliberately cultivated by humans. The techniques developed for cultivating shiitake, growing them on logs inoculated with mushroom spawn, represented a sophisticated understanding of fungal biology, and these methods are still used with modifications in mushroom cultivation today. In ancient Greece and Rome, mushrooms were eaten as food, though with some caution, as Mediterranean regions are home to several deadly poisonous species. The Roman Emperor Claudius was allegedly killed by his wife Agrippina with poisonous mushrooms, hidden in a dish of his favorite edible species. Whether this story is true or legend, it reflects the real danger of mushroom poisoning and the knowledge that while some mushrooms were delicious, others were deadly. The Romans particularly prized Caesar's mushroom, a species still popular in parts of Europe today, with an orange cap and yellow gills. It was supposedly a favorite of the emperors, hence its common name. The Romans developed methods for preserving mushrooms, understanding that while fresh mushrooms were seasonal, they could be dried or preserved in various ways to be available year-round. In many indigenous cultures around the world, certain mushrooms played roles in religious and spiritual practices. In Mesoamerica, several species of mushrooms that contain psychoactive compounds were used in ritual contexts for perhaps thousands of years before European contact. Stone sculptures found in Guatemala and Mexico, dating back as far as 3000 BCE, appear to depict mushrooms and are interpreted by some researchers as evidence of early ceremonial mushroom use. When Spanish conquistadors and missionaries arrived in the New World, they documented indigenous use of these mushrooms, though they often condemned the practices as demonic. Despite suppression during the colonial period, traditional use of these mushrooms persisted in some indigenous communities, and knowledge of these practices was eventually shared with Western researchers in the 1950s, sparking scientific interest in psychoactive mushrooms. In Northern Europe, the fly agaric mushroom, with its iconic red cap and white spots, has a long history of association with folklore and possibly with shamanic practices. Some researchers have suggested that this mushroom may have been used by Siberian shamans, though the evidence is debated. What is clear is that the fly agaric has been known to Europeans for centuries and has featured in folklore as a magical or fairy mushroom. Its distinctive appearance made it a natural symbol of the mysterious and otherworldly, and it appears in countless fairy tale illustrations associated with gnomes, fairies, and woodland magic. In medieval Europe, mushrooms were generally viewed with some suspicion. They appeared seemingly from nowhere, often overnight, grew in rings that were associated with fairies or supernatural forces, and some species were deadly poisonous. The folk names for mushrooms reflect this ambivalent relationship. Devil something or other appears frequently as a descriptor. Deathcap, destroying angel, and similar names warn of dangerous species. But alongside the dangerous mushrooms were the delicious and nutritious ones, and people who learned to tell them apart could supplement their diets substantially. Puffballs, for instance, were gathered when young and white inside, before the spores developed, and eaten as food. Morels, appearing in spring, were highly prized. Field mushrooms, similar to the modern cultivated white button mushroom, were collected from pastures. In various regions, people developed detailed traditional knowledge of local mushroom species, which ones were edible, which were poisonous, when and where each species fruited, how to prepare them for eating or preserving. This traditional ecological knowledge was essential for survival in times when people depended more directly on wild resources. Mushrooms could provide protein and nutrients when other foods were scarce. They were seasonal treats, something to look forward to when the conditions were right. And the knowledge of mushrooms was cultural knowledge, passed from parents to children, part of the fabric of understanding the local landscape. In Eastern Europe particularly, mushroom hunting became and remains a strong cultural tradition. In countries like Poland, Russia, and Ukraine, gathering wild mushrooms is a beloved autumn activity, something families do together, a way of connecting with nature and with tradition. People know the good spots, the productive forests where certain species can be reliably found, and this knowledge is often guarded and passed down within families. The mushrooms gathered are dried for winter use, pickled, or prepared in various traditional dishes. The cultivation of mushrooms began in earnest in France in the 17th century, when farmers near Paris noticed that mushrooms appeared in abandoned quarry tunnels where the soil had been enriched with horse manure. They began deliberately cultivating mushrooms in these caves, and the Paris mushroom, now known as the button mushroom, became a commercial crop. The cool, stable conditions of the quarries proved ideal for mushroom cultivation, and the practice spread, eventually becoming an industry. By the 19th and 20th centuries, mushroom cultivation had become more sophisticated. People learned to produce mushroom spawn, mycelium growing on a substrate that could be used to inoculate growing media. They developed methods for growing mushrooms at scale in controlled conditions. Today, several species of mushrooms are widely cultivated, including button mushrooms, oyster mushrooms, shiitake, and others, making mushrooms available year-round rather than only seasonally. But while cultivation made mushrooms more accessible, it didn't diminish the appeal of wild mushroom hunting for many people. In fact, in recent decades, interest in wild mushrooms has grown, with more people learning to identify and gather edible species, with chefs featuring wild mushrooms in their cooking, and with a growing appreciation for the diversity and culinary possibilities of wild fungi. Mushroom hunting clubs, mycological societies, and educational programs have proliferated, sharing knowledge about identification, ecology, and sustainable harvesting. Scientific study of mushrooms developed alongside these practical and cultural relationships. Early botanists classified mushrooms as plants, not yet understanding that they represented a separate kingdom of life. As microscopes improved and biological understanding deepened, researchers realized that fungi were fundamentally different from plants, lacking chlorophyll, obtaining nutrition differently, possessing cell walls made of chitin rather than cellulose. The science of mycology emerged as a distinct field, and researchers began to document and classify the enormous diversity of fungi. They studied life cycles, ecology, genetics, biochemistry. They discovered the vast underground networks of mycelium, the partnerships between fungi and plant roots, the crucial roles fungi play in ecosystems. Much of this understanding has come surprisingly recently. The recognition that mycorrhizal relationships are nearly universal in terrestrial plants, for instance, only became clear in the later 20th century. Research into mushrooms has revealed their remarkable properties and potential uses. Some mushrooms produce compounds with antibiotic properties. Penicillin, the first widely used antibiotic, is produced by a mold, a type of fungus, and its discovery revolutionized medicine. Other mushrooms produce compounds being investigated for potential use against cancer, viruses, and other diseases. The shiitake mushroom, for example, contains lentinin, a compound that has been studied for its potential immune system effects. Mushrooms are also being investigated for their potential in bioremediation, the use of organisms to clean up pollution. Some mushrooms can break down petroleum products, plastics, and other pollutants that are difficult to decompose. Mycelium can be used to create biodegradable packaging materials, construction materials, and even leather-like textiles. Companies are exploring fungal mycelium as a sustainable alternative to materials like styrofoam and animal leather, growing it into specific shapes using molds, then drying and treating it to create functional products. The science of mycology has transformed from an overlooked branch of botany into a vibrant field addressing global challenges. Researchers have demonstrated that fungi generate value through improved resource efficiency, reducing pollution and greenhouse gas emissions. The transition from chemical processes to biological ones using fungal enzymes has benefited industries including textiles, leather, paper, and food production. This progress built upon generations of basic research into fungal biodiversity, physiology, genetics, and ecology. Today's mycologists work at the intersection of many disciplines. The field has grown from an underappreciated subset of botany to a valuable modern scientific discipline, with significant contributions to science, technology, and industry. Medical mycologists investigate fungal diseases and develop treatments. Agricultural mycologists study plant diseases and beneficial fungal partnerships with crops. Industrial mycologists work on applications ranging from enzyme production to biomaterials. Environmental mycologists examine fungi's roles in ecosystems and climate regulation. The potential applications continue to expand. Recent workshops have explored fungi's connections to climate change, their threats to human health and agriculture, and opportunities for novel medicines and enzymes. Fungi are being studied for their ability to produce biofuels, to remediate polluted environments, and to create sustainable materials. Their enzymes are being harnessed for industrial processes. Their medicinal compounds are being investigated as potential treatments for cancer and other diseases. Throughout all this human interaction with mushrooms, throughout millennia of use and centuries of scientific study, one thing remains constant. Mushrooms continue their quiet existence, fruiting when conditions are right, releasing their spores, contributing to the slow cycles of growth and decay that sustain ecosystems. Our growing understanding of them, our increasing appreciation for their importance, doesn't change what they are. It simply opens our eyes to what has always been there, to the remarkable organisms that have been quietly doing their essential work since long before humans appeared, and that will continue long after we're gone. The quiet science of mycology. There is something inherently meditative about the study of mushrooms, something that slows you down and teaches patience. Unlike many branches of science that move at breakneck speed, mycology often requires you to wait, to watch, to observe processes that unfold over days, weeks, or years. A mycologist studying wood decay might monitor the same log for months, watching as different fungal species colonize it in succession, noting subtle changes in texture and color. Someone researching truffle ecology might visit the same research plots year after year, documenting which trees develop truffle associations and when, building understanding gradually through patient observation. This slow pace isn't a limitation but rather reflects the nature of fungi themselves. Many mushroom species fruit only under very specific conditions, perhaps appearing for just a few weeks each year when temperature, moisture, and other factors align perfectly. If you want to study a particular species, you must learn when and where to find it, then return at the right time year after year. You develop a relationship with specific locations, specific forests, visiting them through the seasons, learning their rhythms. Mushroom identification itself is an exercise in careful observation. You can't identify most mushrooms from a glance. You need to notice cap color, texture, and shape. You need to look at the gills or pores underneath, noting their color, spacing, and how they attach to the stem. You examine the stem, checking whether it's smooth or scaly, hollow or solid, whether it has a ring or vulva. You might need to cut the mushroom and watch whether the flesh changes color. You might need to note the smell, which can range from pleasant fruity scents to chemical or fishy odors. You might even need to examine spore color by making a spore print, letting the mushroom drop spores onto paper overnight to see what color they are. All of this requires slowing down, being present, paying attention to details. In our fast-paced world, there's something deeply calming about this necessity for patience and careful observation. You can't rush mushroom identification. You need to be methodical, thorough, present with what you're observing. And even with all this careful observation, identification is often difficult. Many species look similar. Keys and guides help, but they require working through characteristics systematically, comparing your specimen against descriptions, ruling out possibilities until you narrow down to the most likely identification. Sometimes you need a microscope to examine spores or other microscopic features. Sometimes even experts struggle with identification, needing to consult specialists or even using DNA analysis to be certain of a species. This difficulty, this complexity, is part of what makes mycology fascinating. There are estimated to be between 2.2 and 3.8 million species of fungi on Earth, and we've formally described only around 150,000 of them. That means the vast majority of fungal species remain unknown to science, waiting to be discovered and named. Every mushroom hunter has the potential to find something rare or even unknown, something that hasn't been documented in their region, or perhaps something entirely new to science. Research into fungal intelligence has revealed surprising capabilities. Fungi can solve maze puzzles, growing their mycelium through a maze to find the shortest path to a food source. They can anticipate periodic events, responding to regular environmental rhythms. They can make decisions about where to allocate resources, effectively optimizing their growth patterns to maximize efficiency. These aren't conscious decisions in the way we think about them, but they're sophisticated responses that emerge from the fungal network's collective behavior. The mechanism behind these capabilities involves the mycelial network processing information through its structure. When one part of the network encounters a resource or a threat, chemical and electrical signals propagate through the network, influencing growth patterns throughout. The network can integrate information from many sources simultaneously, responding to gradients of nutrients, moisture, and other factors in ways that optimize the fungus's chances of survival and reproduction. Studying these phenomena requires patience. You might set up experiments where fungi grow through artificial environments, tracking their growth patterns over days or weeks. You might monitor how they respond to different stimuli, to nutrients placed at different locations, to barriers they need to grow around. The work is often repetitive, requiring careful documentation and measurement, but it reveals the remarkable capabilities of these organisms that grow so slowly we can barely see them moving. Some mycologists study extremely slow-growing species, organisms that might add just millimeters to their mycelial network each year. Arctic and alpine fungi, growing in extreme environments where the growing season might be just a few weeks long, can take years or even decades to accumulate enough resources to produce a single mushroom. Studying these species requires extraordinary patience, monitoring the same sites year after year, sometimes with long periods where nothing visible happens, all to understand how these fungi survive in such challenging conditions. The discovery of new species often happens through this kind of patient work. A mycologist might notice something unusual, a mushroom that doesn't quite match any known species. They collect it carefully, document its characteristics, perhaps photograph it in situ. Back in the lab, they examine it microscopically, measuring spores, looking at tissue structure. They might extract DNA and compare it to known species. If it appears to be new, they write a formal description, comparing it to similar species, explaining what makes it distinctive, giving it a scientific name. This process can take months or years from initial discovery to publication. Mushroom conservation has become an increasingly important part of mycology. As habitats are destroyed or degraded, fungal species are lost, often before we even know they existed. Some mushrooms are specialists, requiring very specific conditions or associations with particular plant species. When those conditions disappear, the fungi disappear too. Mycologists work to document fungal diversity before it's lost, to identify threatened species, to understand what conditions they need to survive. This work often involves visiting the same sites repeatedly over many years, documenting which species appear, when they appear, how abundantly they fruit. It's work that requires dedication, a willingness to commit to long-term observation, to build knowledge gradually through accumulated experience. It's not flashy or dramatic. It's quiet, patient work, but it's essential for understanding and protecting fungal diversity. The tradition of mushroom foraging connects amateur enthusiasts with this scientific mindset. Serious mushroom hunters learn to observe carefully, to note details, to build their knowledge gradually through experience. They keep records of when and where different species fruit, building their own local knowledge of fungal phenology. They learn to recognize habitats where certain species are likely to appear. They develop a relationship with particular forests, visiting them regularly through the seasons. This practice of regular observation, of patient learning, of building relationship with place, has much in common with scientific study. The best mushroom foragers develop an almost intuitive understanding of fungal ecology, knowing without thinking about it where to look for certain species, when conditions are right for fruiting, what the presence of certain mushrooms suggests about the forest ecosystem. There's a meditative quality to walking through the forest looking for mushrooms. Your attention focuses downward and outward simultaneously, scanning the forest floor and the bases of trees, alert for the shapes and colors of mushrooms, but also relaxed, moving slowly, allowing yourself to notice. Time seems to slow down. The concerns of daily life fade as you become absorbed in the search, in the quiet pleasure of finding a beautiful specimen, in the satisfaction of successful identification. Many people who take up mushroom hunting report that it changes how they experience nature. They become more observant, noticing details they would have overlooked before. They develop a deeper appreciation for the complexity of forest ecosystems, for the hidden processes happening all around. They find peace in the practice, in the slow walking through forests, in the focused observation, in the accumulation of knowledge and experience. Scientific research into mushrooms continues to reveal surprises. Despite centuries of study, despite the enormous progress mycology has made, we're still discovering fundamental things about how fungi live, how they interact with other organisms, what they're capable of. The mycelial networks linking trees were only recognized and studied seriously in recent decades. The extent of fungal diversity continues to surprise researchers. New species are described constantly, and genomic studies keep revealing that species we thought we knew well are actually complexes of multiple species. This sense that there's always more to discover, that patient observation will reveal new wonders, makes mycology endlessly fascinating. You can study mushrooms for a lifetime and still encounter species you've never seen before, still learn new things, still find mysteries to investigate. The slow pace of fungi, their hidden lifestyles, their enormous diversity, all contribute to this sense of endless possibility. In our modern world that often feels too fast, too loud, too demanding of our attention, there's something healing about the slow pace of mycological study. Whether you're a professional researcher or an amateur enthusiast, engaging with fungi teaches patience. It rewards careful observation. It asks you to slow down, to be present, to notice details. It connects you to cycles and rhythms that are much older and slower than human concerns. And in that connection, many people find a kind of peace, a reminder that there are other ways of being, other speeds of life, playing out quietly all around us. Mushrooms in peaceful places. Picture a misty autumn morning in an old forest. The air is cool and damp, carrying the earthy scent of leaf mold and fungal growth. Light filters through the canopy, creating soft, diffuse illumination that makes everything seem dreamlike, slightly unreal. The forest is quiet except for the occasional drip of condensed mist from leaves, the distant call of a bird, the soft crunch of your footsteps on the path of fallen leaves. This is mushroom weather, mushroom time. Overnight rain has soaked the forest, and the cool morning temperatures are perfect for mushroom growth. As you walk slowly through this peaceful scene, you begin to notice them everywhere. A cluster of honey-colored mushrooms at the base of an oak tree, their caps still glistening with moisture. A line of small brown mushrooms following a buried root through the leaf litter. A magnificent bracket fungus extending from a fallen log, its underside covered in thousands of tiny pores. Each mushroom is a small world unto itself. Kneel down to look closely at a cluster of tiny pin mushrooms, each one perfect in miniature, caps barely 5 millimeters across. Notice how they grow in a specific pattern, spaced evenly, all at the same stage of development, all part of the same mycelial colony beneath the surface. There's something mesmerizing about their uniformity, their delicate structure, the way moisture beads on their caps. Walk deeper into the forest, where old trees tower overhead and the ground is carpeted with moss and needles. Here, in the dimness beneath the conifers, you might find a mushroom straight out of a fairy tale. The fly agaric with its scarlet cap spotted with white. It's large, commanding attention, impossibly vivid against the muted greens and browns of the forest floor. Despite knowing its toxicity, you can't help but appreciate its beauty, its perfect form, the way it seems to glow with color in the forest shadows. Near a rotting stump covered in moss, you discover a shelf of oyster mushrooms, their white or pale gray caps overlapping like tiles on a roof. Their surfaces are smooth and slightly moist, pleasant to touch. They're fresh, just emerged, not yet discovered by the slugs and insects that will eventually colonize them. For this moment, they're perfect, a temporary sculpture growing from decay, transforming dead wood into fungal tissue. In spring, the scene is different but no less peaceful. The forest floor is still damp from snowmelt, and morel mushrooms push up through fallen leaves. Finding morels requires slow walking, careful observation, learning to spot their honeycomb caps among the chaos of sticks and leaves. When you find one, there's a thrill of discovery, a satisfaction in successful search, and usually more nearby, as morels often fruit in groups. Summer brings different mushrooms. In meadows at forest edges, you might find puffballs in the grass, round and white, some as small as marbles, others as large as softballs. Press one gently and it yields, firm but giving like a dense foam. Later in summer, these same puffballs will turn brown and papery, filled with billions of spores, puffing clouds of brown dust when disturbed. In damp areas near streams, you might encounter mushrooms in colors you didn't know fungi could produce. Bright orange waxy caps, their surfaces sticky and gleaming. Purple Cortinarius species with caps the color of amethyst. Tiny red mushrooms like drops of blood on mosses. Each one a surprise, a splash of unexpected color in the forest. Gardens can be mushroom habitat too, especially if there's wood chips or compost, dead wood or old roots beneath the soil. After warm summer rains, you might wake to find your garden suddenly populated with mushrooms. Small brown ones in the lawn, larger ones near shrubs, ink caps that will liquefy by afternoon. These garden mushrooms are doing the same work as their forest cousins, breaking down organic matter, cycling nutrients, connecting with the roots of your garden plants through mycorrhizal relationships. Even in urban environments, mushrooms appear in surprising places. Tree stumps in parks develop shelves of turkey tail fungi, their concentric zones of color making them look like abstract art. Mulched flower beds produce mushrooms after rain. Old trees host polypores, some species persisting for years, slowly growing larger. These urban mushrooms are reminders that nature persists everywhere, that the processes of growth and decay continue regardless of human construction and activity. The sensory experience of encountering mushrooms is part of their appeal. There's the visual beauty, of course, the endless variety of forms and colors. But there's also texture, the feel of a smooth cap, the waxy quality of some species, the spongy firmness of others. There are scents, ranging from pleasant fruity or anise-like fragrances to the earthy smell of mushroom flesh to occasionally unpleasant chemical or rotting odors. Some mushrooms have distinctive sounds. Tap a giant puffball and it sounds hollow, drum-like. The flesh of some species makes a squeaking sound when fresh. The spore mass inside mature puffballs produces a hissing whisper when released. These subtle sounds add to the experience of discovery, making the encounter with each mushroom more multisensory, more memorable. The taste of edible mushrooms varies enormously. Sweet, nutty chanterelles, meaty, umami-rich shiitake, delicate oyster mushrooms with their subtle flavor, the intense aromatics of matsutake. Each species has its own flavor profile, its own culinary possibilities. But beyond the edible species, which should only be eaten after certain identification, all mushrooms participate in the same essential processes. All contribute to the functioning of their ecosystems. Photographing mushrooms has become a popular hobby, combining the pleasures of mushroom hunting with artistic expression. A mushroom makes a perfect subject. It stays still. It often has beautiful form and color. It presents well at close range. Photographers learn to work with natural light to capture the texture of a mushroom surface, to show its habitat context. The best mushroom photographs don't just document but evoke, capturing something of the atmosphere, the sense of place, the quiet beauty of these organisms. The experience of finding a particularly rare or beautiful mushroom creates lasting memories. Perhaps a cluster of golden chanterelles glowing in a beam of sunlight. A massive shelf of sulfur shelf fungi erupting from an oak trunk. A fairy ring of mushrooms in a meadow. Dozens of individuals in a perfect circle. These encounters become stories you tell, moments that stand out, instances where the patient practice of observation was rewarded with something special. For many people, time spent looking for mushrooms becomes a form of moving meditation. The activity requires enough focus to keep your mind occupied, scanning the forest floor, processing what you see, but it's not so demanding that it creates stress. Your body moves at a comfortable pace. Your breathing slows and deepens. Concerns about work or relationships or whatever else might be troubling you fade into the background as you become absorbed in the search, in the pleasure of being in nature, in the accumulation of small discoveries. The mushrooms themselves embody a kind of peace. They don't struggle or fight. They simply grow slowly and steadily, responding to conditions, producing fruiting bodies when the time is right, then fading away. Their lifestyle is one of patient persistence, of existing in harmony with their environment, of performing their ecological role without drama or conflict. Observing them, learning about them, searching for them connects us to that peaceful way of being. In Japanese culture, there's a concept called shinrin-yoku, or forest bathing, the practice of spending time in forests for health and well-being. Research has shown that time in forests reduces stress, lowers blood pressure, improves mood. The practice doesn't require any particular activity, just being present in the forest environment, taking it in through all your senses. Mushroom hunting is a natural accompaniment to forest bathing, giving you a focus for your attention while allowing you to absorb all the benefits of forest time. As evening approaches in the forest, and the quality of light changes to something golden and slanting, the mushrooms take on a different character. Backlit by low sun, a translucent mushroom cap might glow, revealing its internal structure. Long shadows emphasize textures and forms. Colors seem to deepen and intensify. It's a magic hour for mushrooms, a time when they seem most like inhabitants of fairy tales, most likely to be doorways to other worlds. And at night, in forests where bioluminescent species grow, there's a different magic entirely. Those glowing mushrooms create tiny points of green light in the darkness, enough to navigate by if your eyes are dark adapted, enough to transform the forest floor into something like a star field. Standing in such a forest on a moonless night, surrounded by these quiet, glowing organisms, time seems to stop. The ordinary world falls away. There's only the darkness, the soft green glow, the sense of being present in something ancient and strange and beautiful. These peaceful encounters with mushrooms, whether in deep forest or suburban garden, whether seeking rare species or simply noticing the mushrooms that appear after rain, offer something valuable. A connection to natural processes, to cycles that continue regardless of human activity, to forms of life that have their own ways of being. In paying attention to mushrooms, we step outside our usual concerns, we slow down, we become more observant, more present. And in that presence, we often find a kind of peace that's increasingly rare in modern life. As we come to the end of our quiet journey through the world of mushrooms, let's take a moment to reflect on what we've discovered together. We've traveled from the deep past, from ancient aquatic fungi over 1 billion years ago, through the colonization of land and the coevolution of fungi with plants, through human history and our long relationship with these remarkable organisms, to the present day where we're still discovering new species and new capabilities in fungi we thought we understood. We've explored the hidden networks beneath the forest floor, those vast mycelial connections that link trees and plants in ways we're only beginning to fully understand. We've marveled at the diversity of mushroom forms, at the endless variety of shapes and colors and strategies that fungi have evolved. We've considered their essential ecological roles, how they make possible the cycling of nutrients, the growth of forests, the functioning of almost every terrestrial ecosystem. We've appreciated their beauty, their strangeness, their quiet persistence. Through all of this, perhaps what emerges most strongly is a sense of the slowness of fungal time, the patience of fungal existence. Mushrooms don't rush. They grow when conditions are right. They fruit when the moment comes. The mycelium can wait for years, decades, even centuries, persistent in the soil, ready to respond when opportunity arises. There's wisdom in this patience, in this willingness to exist in harmony with natural cycles, to persist without forcing, to thrive by adaptation rather than resistance. In learning about mushrooms, we learn different ways of being in the world. We're reminded that success doesn't always mean being the fastest or the loudest or the most visible. Sometimes success means being patient, persistent, connected, adaptable. It means doing essential work quietly, without fanfare, without demanding recognition. It means being part of larger patterns and cycles, contributing to systems that are much greater than any individual. These quiet lessons from the fungal world might be exactly what we need in our busy, fast-paced, often stressful lives. The mushrooms invite us to slow down, to observe carefully, to appreciate details, to find beauty in subtle things. They remind us that there are other timescales than our daily deadlines, other concerns than our immediate worries, other patterns than the ones that dominate our attention. So as you drift towards sleep tonight, imagine yourself in a peaceful forest, surrounded by the quiet presence of mushrooms in their infinite variety. Feel the coolness of the forest air, the softness of the moss beneath your feet, the peace of being in a place where time moves differently, where the urgent demands of modern life seem distant and unimportant. Let the mushrooms be there in your mind, growing slowly in the darkness, doing their patient work, connected through vast underground networks, part of cycles that stretch back millions of years and will continue far into the future. Rest in the knowledge that this world continues, that the processes of growth and decay and transformation go on, that the mushrooms will fruit again next season, that the mycelium persists in the soil, that there's a constancy and reliability to these natural patterns that asks nothing of you and offers everything. A reminder of connection, of cycles, of the patient persistence of life. If you've enjoyed this journey into the world of mushrooms, please consider liking this video and subscribing to Deep Science Sleep for more sleepy explorations of the natural world and the fascinating topics that help us understand our planet and our place in it. Sweet dreams, and may you rest as peacefully as a mushroom growing in the forest floor.